Type 2 diabetes, also known as adult onset diabetes, is a metabolic disorder characterised by high blood glucose. Obesity is thought to be the primary cause of type 2 diabetes in people who are genetically predisposed. Thus, overweight people have increased levels of glucose circulating in their blood. The pancreas is an organ which produces hormones that control blood glucose levels. The hormones that control blood glucose are produced by clusters of cells in the pancreas called islets. Here glucose is shown arriving at the islets via the bloodstream. The glucose is taken up by cells in the islet called beta cells. The beta cells respond by producing the hormone insulin, which lowers blood glucose. In type 2 diabetes, high levels of glucose stimulate the beta cells to produce lots of insulin to lower the blood glucose levels. The insulin is taken up by the blood vessels for transport to muscle, liver, and fat, where it allows those tissues to absorb glucose. Unfortunately, sustained production of insulin also leads to secretion of large amounts of another hormone-like substance by the beta cells. White blood cells, which live in the islet, respond to this hormone and consume it. We are now inside an islet white blood cell at a magnification of 250,000 times. The hormone, in combination with high glucose, causes production of free radicals inside the white cell. The free radicals activate a sentinel protein that originally evolved to protect us from microbial infection, causing it to unwind. The newly active sentinel proteins then aggregate with 6 other proteins to form a ring. Seven adaptor proteins then bind to the ring. The adaptor proteins work to capture specialised inactive proteins from the surrounding cell fluid, bringing 2 halves together to form the active whole, which is then released. This entire structure is called the inflammasome, and its sole purpose is to act as a platform for combining and activating these proteins. The newly active proteins then convert yet another small signalling protein, splitting it into its active form. This protein is the key protein involved in inflammation in the body. The inflammatory protein is then secreted from the white blood cells into the islet. Unfortunately, this inflammatory protein causes death of the beta cells. Over time, beta cell death causes the islets to shrink, accelerating disease progression. As the islets are now greatly reduced in size, they cannot produce enough insulin to control blood glucose levels. Over time, high blood glucose causes damage to many different tissues throughout the body. What originally evolved as a defence mechanism against infection now seems to be a driving force for inflammation in type 2 diabetes. Chronic diseases associated with inflammation are the most common diseases of ageing in the Western world and represent our greatest health threats in the 21st century. Australia is facing an increasing epidemic of type 2 diabetes. There are now approximately 1 million people living with diabetes and around 100,000 new diagnoses each year. Obesity is thought to be the primary cause of type 2 diabetes in people who are genetically predisposed. In part 1, we explained how inflammation in type 2 diabetes destroys the pancreas tissue and dramatically decreases insulin production. Here we describe the insulin receptor in type 2 diabetes.
What we have discovered is the molecular detail of how the hormone insulin binds to its so-called receptor protein on the surface of a cell and instructs that cell to take up glucose from the blood and to store it as an energy source. The receptor itself actually spans the cell membrane, so it has a part outside, a part inside, and insulin binds to the part that is outside. Insulin is a small molecule whose structure was determined a long time ago in 1969, but it's been unknown how it interacts with the receptor, and that is what we have discovered. And its interaction I've characterized as a molecular handshake as the two proteins dock together. When insulin binds to the outside parts of the receptor, it brings the two halves of the receptor together, and at the same time, the two parts inside of the cell unwind and make contact. These two proteins then activate each other.
This activation allows the insulin signal to enter the cell. Specialised proteins interact with the activated receptor just below the cell surface. The receptor activates these proteins, and they in turn are able to activate multiple other proteins in a long signal cascade. This complex signalling process eventually tells the cell to take up glucose for use as an energy source. The long signal cascade allows tight control of the signalling process, as it can be turned on or off at many different points along the way. In type 2 diabetes, we believe insulin binds to its receptor normally, but the signal is not sent into the cell and the signal cascade does not develop. This is known as insulin resistance. This means that glucose cannot enter the cell and blood glucose remains high. Over time, high glucose levels damage many different organs.
Insulin, as you know, has been used to treat diabetes for a very long time. But all of those formulations and types of insulin that have been used in the diabetes context have been designed and formulated without any knowledge of the way in which insulin actually interacts with the receptor. And now for the first time we've got a picture of that, and we can use that into the design of new insulins that could be used for the treatment of diabetes. So we could imagine, for example, the design of insulins that act for a longer period of time, design of insulins that act more quickly, design of insulins that are more stable upon storage, and that's where we'd like to go with our discovery.